Welcome to the first main lecture in DIY transcriptomics. In this lecture, we're going to set up your laptop with the computing environment that you'll need to be successful throughout the course and that will continue to work for you long after the course is over. If you head on over to the course website and click on the lecture card for today, you'll be taken to a page that contains all the information you need for today's lecture, including a link to the very slides that you're looking at right now. Let's take just a minute to talk about the learning objectives. We want to give a brief overview of the tools that you'll be using throughout the course, including the R programming language, bioconductor, and how these two are related but distinct, RStudio, and more. We also want to actually set your laptop up with all the software that you need for the course. And in most cases, I'll demonstrate the installation of each piece of software directly in this video. So what is it we'll actually be installing? Well, as you might guess, we're going to install the R programming language, but having that programming language in and of itself is really not a great way to actually work in R. So we'll also install RStudio, which many of you are probably familiar with. Its popularity has grown tremendously in recent years. And it's what we call an IDE, or integrated development environment. It gives us a really sophisticated and powerful user interface and user experience for working in the R programming language. We'll also install two text editors, Sublime and Visual Studio, or VS Code. Although these are text editors, they're much more powerful than the traditional text editors you're probably familiar with from what comes installed in your operating system. They're both code aware, so they will recognize R code versus Python code, and they provide a lot of useful features anytime we're working with command line tools and lots of text files. I also want to highlight that Visual Studio Code is actually far more than just a text editor. We're going to use it at the end of the course to connect remotely to GitHub repositories so that we can push and pull projects from GitHub. And then finally, you're going to install Conda, which is a package and an environment manager. Don't worry if you don't know what that means. Suffice it to say, having Conda on your computer will open your computer up to a whole world of bioinformatics tools that you will much more easily be able to install on your laptop. And then finally, I just want to highlight that today's lecture is paired with an in-person lab, and that lab is optional, but the main goal of the lab is to provide IT support for anyone who struggles to get any of that software that I just described installed on your computer. So you can come in and get one-on-one help from myself and the TAs. You may have noticed that I did not mention Excel in that list of software, but that's not to say that Excel is not an important tool in the data scientist toolbox. It absolutely is. But at its heart, Excel is accounting software. And so when you give it gene expression data, it can do some funny things, and we just want to be aware of that so that when we use Excel, we know what we're getting. Perhaps one of the best examples of how Excel can cause problems with gene expression data comes when we think about gene symbols that look like dates. So that includes actually quite a lot of gene symbols and actually entire families of genes, like all of the dectins that start with DEC. Those get treated as December, the septins as September, and a group of membrane-associated ring finger proteins, or marches, as the month of March. And unless you know to look for these, it's virtually impossible to spot them because, after all, we're working with a very large spreadsheet, so you don't notice that Excel has corrupted these gene symbols and turned them into dates. It's also not easy to turn them back, and so it poses a real problem if you're opening up large gene expression studies or data sets in Excel. You want to be aware of this. I find it remarkable, but every few years a new paper comes out on exactly this same topic. Here are two examples, essentially just saying that this is an issue that's widespread. We see increasing numbers of supplementary files with mistaken gene symbols that have been corrupted by Excel. This is likely due not to people making more mistakes, but just the fact that as we've gone through the last decade or so, we're seeing a tremendous increase in the amount of sequencing data in general, and of course a lot of that ends up in supplementary files. And you can see that there's really no particular journal that's spared from this. In fact, some of the most prestigious journals are also the most egregious in terms of the prevalence of gene name errors. So I didn't actually explain why we chose the R programming language for this class. And to be honest, it's the language that I learned first, and it's the one that I feel by far the most comfortable working in. But that's not to say that there aren't other programming languages you may want to explore, or that could be as good or better, frankly, for doing some of the things that we're going to do in this class, and even that you may want to do down the road with biomedical data sets. So I just want to highlight some of these languages. They're ones I'm sure many of you have heard of. Of course, Python is another programming language that's often the preferred choice for biologists. There are many, many tools available in Python for a wide range of biomedical data sets. Rust is a newer language that has garnered a lot of attention. Perl has been around for a long time and is still quite popular for parsing large text files. C++ when you need a very fast programming language, and far more, tools like SQL for database queries. But for the purposes of this class, we will focus entirely on R and the Bioconductor suite of software packages for R. But we will at times leave R and move into the command line to run programs that were written in these other languages. And for that, we'll open the shell or terminal or bash. It's called different things by different people on different computers. And that's really the space that we'll be working in this course. Okay, just a quick note about my computer. I'll be using a laptop throughout the course, and that laptop has changed over the years from an 8 GB machine to now 32 gigs, from the Catalina version of the Mac OS to now Monterey. And I only highlight this because your hardware and software configuration on your own computer is quite likely different than what I'm showing you here, and that's totally fine. It just means that as we move ahead in the next few slides to begin installing software and then using it very soon, you may have an experience that's slightly different than what I'm showing you in the videos. That's okay. It may run faster, it may run slower for you. All of those things are to be expected if your hardware and software configuration is different. I also just want to point out that laptops have come a long way. A 32 gig laptop can do things that we just didn't think of doing on laptops 5 or 10 years ago. Now, although we'll be using our laptop for the entirety of the course, I want to take just a minute to highlight some of the other compute options that are out there. Because to be honest, at some point you may want to do some compute intensive work that your laptop will just not be cut out for. And so that's when it's really useful to turn to one of these other compute options. So what are those options? Well, option one is really simple. It's a lab workstation. So a desktop computer that's more powerful than your laptop, more RAM, more hard drive storage. It has some really nice advantages. The first, and maybe the most obvious, is it's your computer. So therefore, you have unrestricted access. You don't have to go through anyone else in order to access this computer. And because it was purchased for your group, your lab, your company, it presumably has all the software you need. Somebody will set that up because it's going to be tailored really specifically to meet your needs as a lab or as a group. It's also going to be quite secure because it will be set up in a way that it's behind your institutional firewall. Option one is not without its disadvantages, and the first, maybe the most obvious, is there's got to be some initial capital investment. Somebody has to buy this computer, and depending on the configuration, that cost can be quite high. Somebody also needs to serve as the systems administrator for this machine. In other words, they need to be able to maintain and keep the machine working well for everybody in the group. If somebody needs new software installed or updated, the sysadmin would be the one to do that. And then, although I don't view this as a con, some people might, high-end lab workstations often need to, or often are, have a Linux operating system. I love Linux operating systems, but if you're not familiar with them, it can be just another technological hurdle or obstacle for you and your group to overcome. Okay, now what does that amount to in terms of actual investment? Well, I just put up an example here. You know, a 12-core CPU machine with 500 gigs of RAM, 10 terabytes of storage, hard drive storage, could cost you between 10 and 20,000 US dollars. I know that's a lot of money in the US. Labs spend this much on many smaller pieces of equipment, so I think this is a pretty good deal for the price for what you get, given how often this kind of computer could be useful for the broader group's research, but that's just my opinion. Option two is the cloud. In other words, somebody else's computer, and that's often Amazon or Google, since they are the biggest suppliers of cloud computing infrastructure. This has some nice advantages. One is that it's scalable based on your needs, right? Over here in option one, if you end up wanting to do something that takes more RAM than your computer has, there's no adapting to that. You can't use that computer. But with option two, you can scale your compute resources based on what you need. It's also very easy to set up. You use a web interface to click around and configure the computer, and that tells Amazon and Google exactly what kind of machine in the cloud they should set up for you. And then finally, software installation has been made much easier on these cloud instances or computers by virtue of Docker and Conda. And we'll talk about Conda again in just a few minutes. But there are some really, I think, important cons to highlight here. One is that depending on your view of cloud computing infrastructure, this may not be the level of security you need. Certainly, if you're working at a government institution, you don't just throw your data on Google or Amazon. It can also be challenging in terms of data transfer. Anytime you want to do an analysis, your raw data has to be moved to that cloud computer, and then any outputs need to be moved back to your own computer, and that can take time. And then finally, you can accidentally accrue charges. In general, cloud computing can be expensive, but you can also unintentionally accrue charges if you leave your computer up and running. Okay, and in option three is a compute cluster. If you're at a large university or a large company, there's a good chance that your institution has a powerful compute cluster. And that's great because those are also scalable based on your needs, just like cloud computing. And they're highly cost efficient because you can ask for just the amount of resources you need. You don't have to worry about accidentally leaving your cloud computer running. The systems administrator is taken care of here in this case. This might be the single biggest advantage because you don't have to be an expert then on maintaining the Linux machine. Your sysadmin does it for you. And then it's of course secure because it's behind that same institutional firewall. There are cons associated with a compute cluster. You actually may have to frequently request to have software installed or updated. They oftentimes have a large group of individuals to manage and to provide their services to. So sometimes that means it can take a while before the sysadmin or sysadmin group for that compute cluster can get around to updating or installing the software you need. And there may actually be pieces of software that they deem inappropriate or unable to be safely administered on a compute cluster. There can also be a steep learning curve. You have to actually send jobs to the cluster, and that requires a bit more command line savvy that some people just may not have right out of the gate. And then lastly, oftentimes these servers, because of their size, complexity, they can have frequent and unannounced server downtime, which can be a nuisance when you're trying to run jobs and get work done. I feel that I'd be remiss if I didn't at least mention that in addition to the whole R bioconductor world that we'll be working in this course, there's this other world of commercial software solutions that include things like CLC Workbench, GeneSpring, IPA, Partech Genius. But I want to be clear, we won't be using or talking about any of these pieces of software at any point in the course. And that's because personally, I feel they have a number of important drawbacks to be aware of. The first is that they're really expensive. They almost all follow a subscription model, which means you have to pay thousands, sometimes over $10,000 per year to keep using the software. They're also not open source, and I'm a big proponent of open source software. Because they're not open source, oftentimes you don't really know exactly how an analysis is done or how a particular figure was generated. I also think there's real issues with reproducibility. These are all point and click graphical applications, and it's really hard to reproduce a series of cursor movements and mouse clicks. And then I feel there's this inherent trade-off between power and simplicity. Software can be really simple or it can be powerful, but it's hard to be both. And these software applications, by and large, try to be very powerful. They want to offer their users a wide range of things that they can do with a wide range of different data types. And as a result, they're not simple to use, oftentimes requiring that you watch lots of different tutorials or read help manuals, and that can take a lot of time. If I'm going to talk about the pros and cons of commercial software, it's only fair that I do the same for what we'll be using in the course, R and bioconductor. Maybe the biggest pro of using R in Bioconductor is that it's free and open source. You will never have to pay anything to use R. It's also been used for over a decade to analyze genomic data. So there's a rich tradition and a rich set of tools for a wide range of genomic data types. You also often don't even need to write code from scratch, de novo. And that's because chances are the question you're asking is a question that other people before you have wanted to ask. In which case, there's code, perhaps extensive code, already out there that you can find. And because it's open source, you can use it. And that oftentimes can get you 50, 60, 70, 80, 90% of the way. You can adapt that code then to do exactly what you want to do with exactly your specific data. There's also a huge user community for R, which is so important for continuing learning and also when you need troubleshooting because you've run into some issues. R produces publication quality graphics, so you don't need to take a plot produced by R and then spend hours making it look good in Illustrator or Photoshop. The results you get with R are reproducible, and we have a whole lecture devoted to reproducibility. It's a really important topic in bioinformatics, so we'll talk a lot about this topic later in the course. R, by definition, is modular and standardized in the way you work in the R programming language, and we'll cover this kind of topic later. And then finally, it's not just for transcriptomics data. This course focuses on RNA-seq because it's a convenient data type to work with that everyone can relate to. But you can use the R programming language and Bioconductor for ChIP-seq data, single-cell RNA-seq, population genetics data, ecology and evolution, genetic screens, interaction networks, machine learning, and frankly, much, much more. But it has its cons as well. There's a steep learning curve. Like any programming language, there's a lot of stuff to learn. My hope is that by the end of this course, we've flattened that curve a lot and you can really take off with your learning. There's also no single right way to do anything, and that's not a feature of R, that's a feature of any programming language. That's often a point of frustration for many people learning R because they want to know what's the right way to analyze RNA-seq data, what's the right way to map reads to a reference genome or transcriptome. And the answer is there isn't a single right way. There's many different tools, for example, for mapping reads to a reference. So we will talk a little bit about the tools that I like to use, but just be aware they're not the only option for doing the things that we'll do. And then finally, historically, R was really lacking in terms of a user interface. That's all changed with RStudio, so I don't really view this as a con anymore. Okay, as we get ready to begin installing all the software we need for the course, I wanted to structure things in a way that would be really, really clear for you. This table summarizes all the pieces of software that we want to install, what they all do, in other words, why would we care to have them on our computer, how you get them, and then I know this is a little silly, but are they free? Are they cross-platform? And it's just all yeses. In other words, everything we're going to install that we'll use for the rest of the course is absolutely free and will work on either a Windows machine or a Mac. Now, I really think of this table as having two sections to it. The first section are the 4 pieces of software at the top. They are all essentially click and install. In other words, you'll follow the link that I've placed here. You'll follow the instructions on the software developer's website for how to download and install them. And I'll actually demonstrate this for you for R and RStudio, and I'll leave you to do Sublime and Visual Studio Code on your own. Again, they all are super easy click and install pieces of software. However, the rest of the table is non-R, non-bioconductor software. These are command line tools that I think are generally useful to have, but they require a bit more effort to install. We will install all of these using Conda. And if you recall from previous videos, Conda is that environment and package management system that has become really popular and really useful for making installation of software and the dependencies that the software has much more pain-free. Now, I will just tell you right now that although I'll demonstrate how to install all these using Conda, Callisto is the most important. It's the piece of software we'll use for mapping reads. So there's a chance you run into issues with other pieces and installing other pieces of this software. And if you do, my best suggestion for you is to just don't worry about it. Forget about it. Don't install it. Callisto is one we want to make sure that everybody has a chance to install. But if you have a trouble installing, for example, Sour Mash, don't worry about it. I will still demonstrate for you how you can use all these pieces of software, why I think they're useful, and why I think they work well on a laptop. Later in the course, actually at the end of the course, we will try to install a version of Callisto, if you will, called Callisto Bus Tools. And it's a Python program that will let us really easily process single cell data directly on our laptop, which is pretty amazing. But if you can't install that now, that's okay. We won't even use this until almost at the end of the course. So you have a whole semester for us to kind of help you get that software installed. So we'll be using Conda to install all of this software, with the most important one being Callisto. And I've provided a link here to a pretty detailed step-by-step recipe for how to do the installation of all of these using Conda, and we'll work through that together. That really wraps up this first video. In the next video, we'll pick right up here and actually get down to the business of installing software.